Today I will be talking about a common biochemistry lab technique known as sodium dodecyl sulfate polyacrylamide gel electrophoresis, or SDS PAGE for short. So I was recently a teaching assistant for an undergrad biochemistry class here at UC Davis, in which the students got some exposure to SDS PAGE. In grading one of the exams, it became clear that there were some significant misunderstandings with the technique and what the gels look like over the course of an experiment. I realized that SDS PAGE, like a lot of other lab techniques, can be difficult to understand if you haven't either done them yourself or watched somebody do them. So I know there are students in these classes that will go on to take a lab class where they do the technique, or will go on to work in a research lab and will do the technique themselves. But some students will not. So I wanted to try to put together a teaching tool to try to help students understand the concepts of SDS PAGE and what the gels look like over the course of an experiment. Alright, so to try to put things into context, I wanted to set up a sort of theme for this teaching tool. So to that end, I would like to purify the enzyme known as rubisco. So rubisco is an enzyme found in chloroplasts. So here I have a sample of chloroplasts from garden pea plants. And rubisco is the enzyme that's involved in the fixation of carbon dioxide. And what you should know about rubisco is that it has two subunits. So there is a large subunit and a small subunit. The large subunit is 50 kilodaltons in size, and the small subunit is 14 kilodaltons in size. So I would like to confirm that the sample indeed contains the rubisco enzyme by checking that the proteins, the large and small subunits, are present. And to do that, I will use SDS PAGE. First, I wanted to talk about some of the goals of SDS PAGE. So one of the goals is to separate proteins based on their molecular weights. Another common goal is to determine whether a complex mixture of proteins contains one or more proteins of interest based on their expected molecular weights. So some of the key concepts of SDS PAGE are that it's a widely used analytical tool for protein separation. The gel itself is made of acrylamide polymers cross-linked with bisacrylamide, and this creates a porous matrix structure, which I've attempted to depict in the bottom right. And on top of the gel are a bunch of wells that samples are loaded into. After samples are loaded, an electric current is applied, and the proteins begin migrating through the gel from the top to the bottom. And over the course of the electrophoresis, proteins are separated from one another based on their ability to move through the pores of the polyacrylamide matrix. And in an SDS PAGE experiment, this is determined strictly on protein size. And so after the experiment, you get separation of proteins based on size, where larger proteins tend to migrate closer to the top of the gel, and smaller proteins migrate closer to the bottom of the gel. Folded proteins have unique sizes and pH-dependent net charges. So how is SDS PAGE able to accomplish protein separation based strictly on protein size? Because in general, the ability of a protein to move through the pores in a gel can be influenced both by protein size and charge. So what SDS PAGE does is it takes advantage of SDS, which is sodium dodecyl sulfate. And this is the structure of SDS. It has a hydrophobic 12-carbon chain, as well as an anionic sulfate head group. And the dodecyl sulfate group interacts strongly with proteins and causes them to unfold or denature, as shown here. So this also first, the anionic sulfate head group also keeps the denatured proteins soluble, so that's also a key quality of SDS. So the denaturation with SDS can also break apart protein complexes into their individual subunits unless there are disulfide bonds between them. And so to try to illustrate that, on the bottom left here, I have a polypeptide chain that has a disulfide bond within the same polypeptide chain, and then below that there's a disulfide bond between two separate polypeptide chains. So the SDS itself does not break the disulfide bonds, but it still denatures the polypeptide. So what you get is, with a single polypeptide, you get this sort of looped structure where the protein is denatured still, but where you have two polypeptides with a disulfide bond between them, they're both denatured, but they're still bonded together.
Okay, you're good.
Okay, so now I'm ready to load my SDS PAGE gel, and I just wanted to show you kind of what these gels look like. So these gels are run in the vertical dimension, kind of unlike an agarose gel, which is run in the horizontal dimension, although this is the same in that you have these wells up at the top of the gel, and these were formed when the gel polymerized around the teeth of the combs. So the first thing that I'm going to do is to remove the comb. Okay. And then now I need to assemble the running apparatus. So now I have to pour in some of the SDS PAGE running buffer. So there's this inner reservoir here and then an outer reservoir. So this just allows the apparatus to carry the current through the system during the electrophoresis. And so these are my chloroplast samples here. So I have 4 of them. And so in the sample loading buffer there are 5 components. So there is tris, which is a buffering agent which maintains the pH. There is SDS, which is used to denature the proteins. Bromophenol blue, which is the dye which allows us to see the movement of the protein samples through the gel during the electrophoresis. Glycerol, which increases the density of the solution and causes the samples to fall to the bottom of the wells. And the last thing that we usually use is a reductant such as beta-mercaptoethanol, and this is used to break disulfide bonds between or within proteins. So to try to sort of illustrate the importance of each of those components, I omitted one or more of those components in these samples. So this first sample here has that complete mixture in the sample buffer. This second sample lacks the beta-mercaptoethanol. The third sample lacks SDS but contains beta-mercaptoethanol, and the fourth sample lacks both SDS and beta-mercaptoethanol. So now I think I'm ready to load the gel. So I'll just get my chair set up. So these, again, I said this, these gels are run in the vertical dimension, unlike an agarose gel, but they're still loaded from the top. So I only have 5 samples, including my molecular weight marker, so that I'll be able to determine the molecular weight of the protein bands that I see on the gel after the electrophoresis. And the lanes that do not have protein samples are just going to be loaded with sample buffer. And hopefully you can see the solution falling quickly to the bottom of the well. So now I'm just going to load the molecular weight marker. I'm going to use a syringe to load the chloroplast protein samples because the volumes are a bit larger. So again, hopefully you can see the sample falling to the bottom of the well and rising up to the top as I load it. So in between samples I'll just wash out the syringe so that I don't carry over any sample from the previous sample into the next lanes. So if you hear any of my lab mates in the background, please excuse them. So I was going to comment on something in one of the samples before our camera battery died, but before that I'm just going to finish loading the gel and then I'll comment on what I saw. So if you notice that the blue dye looks a little bit more diffuse now, it's because I had to clear up some memory on the computer or on the card to make room to record more, and that's why gave the sample some time to spread out a bit. But the last lanes I'm just going to load more sample buffer. There's a little bit of a bubble right there. It's not perfect loading. What I'll do is I'll just use the syringe.
An icebox, just stick it in there. Thank you.
Sometimes some bits of the acrylamide prevent you from loading your samples into the gel. Well, it's not the prettiest gel, but it's a gel nonetheless. So what I notice is in these samples 3 and 4, these are the ones that didn't have the SDS, so there was some precipitate in the bottom of the tube that probably is some insoluble protein because I omitted the SDS. So we'll find out later when we see the result of the gel. So now the gel is fully loaded, I'm ready to start running it. So I'll just grab these lids. Of course it's a tangled mess up here. And I have a pre-programmed cycle for the electrophoresis, so I'll just start. Over the course of the electrophoresis, I took a series of pictures about every 10 minutes to try to show the importance of the bromophenol blue dye that's included in the sample buffer, as well as to show how one determines when the gel is done. So in the top left is the gel before the electrophoresis began, and so all of the samples look like columns of dye. And then after 15 minutes, all of the samples have sort of condensed into bands. And then if you follow from left to right and then go down to the bottom from left to right, you can see that band of dye, which is known as the dye front, it progressively moves further down into the gel. And by the end, by that 55-minute time point at the bottom, the dye has basically left the gel and entered into the running buffer. And so that's how you know when it's done. Once the dye front has reached the bottom, that's a good time to stop running the gel. So now that the electrophoresis is over, I'm going to proceed with staining the gel. So I'll just disassemble this apparatus here. Get this comb here and I will pry off spacer here. Okay, so you can't really see, but before I took the gel apart, I noticed in lanes 4 and 5, which were the samples that did not have SDS, there was a small amount of material that didn't seem to enter the gel, and it's possible that that was protein that was just aggregated because I didn't add any SDS. And so I think the key thing I want you to notice is that you cannot see any protein bands on here yet, so let me put something to contrast. So you can see the molecular weight marker in this lane. So the cool thing about this is the proteins in this sample, they come pre-stained, so you can see the markers without previously staining the gel. But in my chloroplast sample lanes, which are these ones, you don't see anything yet, so that's why I have to stain the gel with Kamasi. So what I'm going to do is just cut away this top part of the gel, which had the wells, and then pry this off, put it in my staining container, and then I will pour some Kamasi Brilliant Blue solution, which is a protein, Kamasi is a protein binding dye, which allows us to visualize the protein bands on the gel. So now the gel has been staining for about an hour, and I'm ready to do the destaining, and I just want to show you why you have to destain the gel. So what I'll do is just pour off the staining solution here to a waste container. And so you can see the gel is dark blue. You can't really see any protein bands on it yet. Now I'll pour some of this on there. Okay. And then I'll just let this go, and this usually takes a few hours, sometimes even overnight. And then that's it. So now the destaining of the gel is complete, so we can visualize the protein bands on the gel. So I just have it here. I got it wet and just put it on some filter paper for a good contrast. So this lane here is the molecular weight marker. So there are various bands of known molecular weight. This lane here were the chloroplast in the sample buffer that had all of the components, so it included the SDS and the beta-mercaptoethanol. This sample here did not contain the beta-mercaptoethanol, and as you can see, it looks pretty similar to the sample that contained the BME. Sometimes there is no difference. It's not too surprising. This sample here contained beta-mercaptoethanol but did not contain SDS. And as you can see, there is a lot less protein that moved into the gel, and it's quite a bit streaky. And then this lane here contained neither SDS nor beta-mercaptoethanol, and virtually nothing entered the gel. So I wanted to comment on the data in a little bit more detail. And what I did is I scanned the gel from the previous video clips and just added a bunch of labels to try to make things more clear. And the first thing I wanted to do was to comment on the differences between the lanes that did and did not have beta-mercaptoethanol. And so if you look at lanes 1 and 2, they both had SDS, but lane 2 did not have beta-mercaptoethanol to reduce any disulfide bonds in the sample. And the band patterns look pretty similar, and a lot of times that's pretty common. So of the proteins that we can see, none of them appear to have disulfide bonds with other proteins. Otherwise, we'd expect to see a difference in their migrations with or without beta-mercaptoethanol. And if you look at lanes 3 and 4, lane 4 just seemed to have less protein overall. And so I guess you can hypothesize yourself about what you think that is. But maybe proteins, when they have disulfide bonds, they for some reason have a higher tendency to become insoluble. So you can hypothesize about that. The other thing I wanted to do was to compare the differences between the lanes that did and did not have SDS. So lanes 3 and 4 were the ones that didn't have SDS, and I briefly mentioned in the other video clip that I think it's pretty obvious that less protein migrates into the gel when you do not add SDS. So remember, I said in the beginning that the SDS, number one, denatures the protein, but the sulfate head group is needed to keep that protein soluble. So the heat of the sample itself will denature the proteins, but the SDS keeps it soluble. So I think when you look at lanes 3 and 4, it seems pretty clear that when you don't add SDS, the proteins have a much lower solubility when they're denatured. And the next thing I wanted to do was to go back to the original question I had, or my goal was to purify the Rubisco enzyme. And I said that it had two subunits, the large and the small, and they were 50 and 14 kilodaltons respectively. And so if we look at the gel, okay, we can see a dark band at right around 50 kilodaltons in pretty much all the lanes, but let's just focus on lane 1 for the rest of this set. So we see a 50 kilodalton band, and then we also see a band that's right around 14 or 15. And so these look like they could be the large and the small subunits of Rubisco, so that's good evidence that I have this enzyme in my sample. But have I proven that my sample indeed contains the Rubisco enzyme? And so remember that SDS PAGE only tells us that we have a protein of a certain size. It doesn't necessarily tell us that, it doesn't tell us the identity of the bands that we see. So oftentimes these experiments are done in parallel with one or more additional experiments, such as Western blotting and immunodetection, where you use antibodies to look for specific proteins, mass spectrometry, where you would cut out a band from a gel, for example, and then use a mass spectrometer to identify the peptides in there and try to map them back to the proteins that you expect, to see which ones are there. And also, importantly, you can do enzyme activity assays. So the sample, remember I just said, I can detect bands of the expected molecular weight, but I want to know if I have a functional protein. And what one might do is to just do an enzyme activity assay. So you know what activity your enzyme is supposed to have, and you can just see if it's able to catalyze the conversion of its substrate to product. And that would also give you further evidence that you purified your protein.